Now let's talk about the plasma membrane functions. The major function, which I've mentioned before, is regulating materials moving into and out of the plasma membrane. The plasma membrane kind of acts like a bouncer. And so if something tries to get through that the plasma membrane doesn't want through, it's going to stop it from getting through. And then those molecules have to find other ways to get into or out of the cell. And so there are different mechanisms of transport that different molecules use. Some of these mechanisms are called passive mechanisms, which means they're not going to use any energy. And then there's active mechanisms, which use energy, typically in the form of ATP, but also in the form of ion gradients. So passive mechanisms, I just mentioned this, don't require energy. The two common types of passive mechanisms are diffusion and osmosis. And there is simple diffusion and then facilitative diffusion, and then osmosis, which we'll talk about. Active mechanisms require energy, either energy of ATP or energy of ion gradients. And so there are different types of active transport mechanisms. One is using a pump mechanism, pushing molecules against a gradient. Another is forming a vesicle around the molecules. So we'll focus on the diffusion or the simple or passive processes first, and then we'll move into the active processes. So diffusion, by definition, is the movement of molecules from higher to lower concentration down a concentration gradient until equilibrium is reached. Think about, no, I think I have a picture. Hold on. There we go. Let's look at this picture here. So think about if this dropper bottle contains some type of a coloring agent, or what if that was filled with Kool-Aid that you put into water and then it disperses out and you have this wonderful drink. So when you depress the lid, depress the squeezer, I don't know, the top, all the solution comes out and it's very concentrated in one area. The molecules in that solution are going to diffuse outwards. They're going to move from higher to lower concentration until we have equal amounts of water to solute ratio all around them. That's what simple diffusion is. Molecules moving from a higher to lower concentration down a concentration gradient across the plasma membrane. And molecules that move through simple diffusion would be your nonpolar small molecules and lipid molecules, small lipid molecules. We do have movement of water, which is a polar molecule that can move across the plasma membrane directly, but water is one of those special molecules. So other than water, polar molecules can't cross the plasma membrane. They're going to need other mechanisms. So here we can see oxygen on one side of a gradient, and on the other side of the gradient we have carbon dioxide. So this would be like our lungs, right, up here, releasing oxygen to our blood. The oxygen is going to move from our lungs and go into our blood. Carbon dioxide, on the other hand, is going to move from our blood and go out into the lungs. Make sense? Facilitative diffusion, then, is the movement of those small molecules that can't move across the plasma membrane regularly or readily. So these are going to be small molecules that are charged, so ions, polar molecules like glucose, There are two facilitative processes that work. Channel-mediated diffusion, where a channel opens and ions can cross, and then carrier-mediated diffusion, which is open to specific molecules that they bind to, and then they move the molecule across. For channel-mediated diffusion, we have some channels that are leak channels, so they're always open, and so ions can cross readily back and forth. There are sodium leak channels, potassium leak channels. These are really important, actually, for our muscle and our nervous system cells. And then we have gated channels. Gated channels are closed, and they only open in response to some stimulus. So these are the ones that are going to open and allow for lots of sodium to rush in or potassium to rush out, and they're going to cause other reactions to occur within the body. So this is channel-mediated diffusion, and these are just leak channels here, just allowing molecules to go in or out of the cell. Carrier-mediated diffusion is where the molecule, like glucose, binds to the protein channel or protein carrier, and then the carrier actually moves the molecule in. So here we have a glucose molecule. Glucose binds to the carrier protein, and then the carrier protein changes in configuration when there's a bound glucose, and it opens to the inside of the cell. So that would be carrier-mediated diffusion. There are different factors that will affect the diffusion rate. So temperature, if you've ever made Kool-Aid, this is a perfect example for anybody who's made Kool-Aid. When you use just ice cold water and you pour the solute, the Kool-Aid packet, into the ice cold water, and your cup of sugar into the ice cold water, and you stir it up, it takes a while for the sugar and the water to diffuse and dissolve into the solution. Whereas if we take the sugar and the Kool-Aid and we add it to a cup of hot water, the sugar and the solute, sugar and the Kool-Aid solute will dissolve very fast because molecules move faster at higher temperatures. And then we can take that hot cup of water and pour it into the rest of the Kool-Aid water, the cold water, and add some ice, and then we have a nice cold glass of Kool-Aid really rapidly, and that's how we typically make Kool-Aid. Steepness of the gradient is going to influence the rate of diffusion as well. If you have only a few sodium molecules on one side of the membrane, then sodium's going to move a lot slower. But if you have thousands of sodium molecules on one side and very few on the other, so say you have 10 on one side and 5 on the other, yeah, sodium will move across, but it's going to move very slowly. You're almost at equilibrium now. But if you have like 1,000 sodium molecule or sodium ions on one side, and on the other side you have 2 sodium ions, sodium is going to move very rapidly across that plasma membrane from higher to lower concentration until equilibrium is met. Osmosis is just a special type of diffusion. So this is the diffusion of water through a selectively permeable membrane. So diffusion, we're typically looking at the solute. With osmosis, we're looking at the solvent or water. And so water moves from higher to lower water concentration. Water passes the plasma membrane. I already mentioned it can move directly across, but more often than not, water is going to move through protein channels called aquaporins. And so the more aquaporins we have, the faster water will move across the membrane. And for water, you're going to always be looking at the amount of water to solute ratio and determining how water is going to move. So if you have a lot of solute on one side of the membrane and less water, water's going to move from where it is to where it isn't. It's always going to follow the solute. One way I always say this is water follows salt. Water always follows salt. So if sodium ions are moving, water's going to move with it typically, okay? And so the movement of water is going to be down a concentration gradient of water from higher to lower concentration until you have equilibrium met. So if you have a 1% solution, or if you have a solution that has 1% solutes and a solution that has 3% solutes, water is going to move from the 1% solutes to the 3% solutes because there's less water to solute ratio with the 3% water or the 3% solutes. And so let me see if I can show you this. All right, so here we have, there's all our water. You can see water over here, but there's very few water molecules. Over here we have a lot of water molecules. Because there's so few water molecules on this side, and we have more water molecules over here, water's going to move from higher to lower water concentration. But it's going to move across the membrane until you have a water to solute ratio on both sides. I like this figure because it kind of shows what's going on. So here we have these large molecules here, these large black dots are representing solutes like glucose, sodium, and proteins. And then all of these tiny red with blue dots, those are the water molecules. So right now we have equal amounts of water on each side. If you count the number of water molecules, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And then you have 3, 4, 5, 6, wait, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. Okay, so there's a few more water molecules on this side, but it's supposed to show roughly the same amount of water on each side. But if we look at the amount of solutes, we have 4 solutes on this side, and we have 1, 2, 3, 4, 5, 6, 7, 8 solutes on this side. So water is going to move from the higher concentration of water to solute ratio to the lower concentration of water to solute ratio until we have equal amounts of water to solute on both sides. And this is how our cells gain and lose water. So osmotic pressure is the pressure that pulls water or keeps water from leaving a cell. So the more solute you have inside of a cell, the higher the osmotic pressure in that cell would be. And so if you have a low osmotic pressure, water is going to move out of the cell very rapidly. And the tonicity of a solution is the ability of a solution to maintain water volume. So there are 3 tonicities. We have isotonic solutions. These are solutions that have the same, relatively same, so similar concentrations of water to solute ratio. So an example, physiological saline has similar amounts of water to solute ratio as our body cells. This is going to be isotonic to erythrocytes. And so if we're going to put water into a person, if we're giving them a solution of water, we're not going to give them distilled water that would cause your cells to take in a ton of water. You're going to give them something that is going to help maintain homeostasis. So you're not going to have a movement of water into or out of the cell. A hypotonic solution is a solution that has lower amounts of solute in the surrounding environment and more solute inside the cell. So in this case, water is going to move from the solution into the cell. And that causes the cell to get very large, and it will burst if it gets too large. That's termed lysis. Hemolysis is rupture of red blood cells. And so we see that right here. And I always think of hypotonic. Sounds like hippo, and hippos are large. They get very large, so cells get large in hypotonic solutions. Hypertonic solutions, the solution has a higher concentration of solutes than the cell itself. And so water is going to move from the cell out into the environment. And so an example, if you have a hypertonic solution, water moves from the cell out into the solution that the cell is in, and the cell will shrivel up. That's termed crenation, and that can also kill the cell. That would dehydrate the cell. And so when I think of hypertonic solutions, the term hyper reminds me of little kids, and little kids are small. And hypertonic solutions are solutions that cause cells to get very small or shrivel up. So here is the figure from your textbook that shows an isotonic solution. You have equal amounts of water moving into and out of the cell. Hypotonic solution, more water is moving in than out. And hypotonic solutions, more water is moving out than in. Wait. There's no video, right? Okay. All right. The next mechanism, or the next thing we're going to talk about, we're going to look at active transport mechanisms. So active transport is the movement that goes against the concentration gradient or moves very large molecules across the plasma membrane. But in active transport, you need some form of energy. So there's primary active transport, which uses the energy of ATP to drive the movement of molecules across the membrane. So sodium potassium pumps use primary active transport. Endocytosis uses primary active transport. So here's an example of an ion pump. I said sodium potassium pump, that's a type of ion pump. Here's a calcium pump. Calcium pumps are going to move calcium from the cell itself out of the cell into the interstitial regions. It moves against the concentration gradient because you're pushing more calcium out than is in the cell. And so that's going to be an active transport mechanism. To activate the pump, you have to use ATP and hydrolyze it to ADP plus the phosphate. And that activates the pump, and the pump pushes the calcium out. The sodium potassium pump is a special pump that binds sodium that's inside of the cell and pushes it outwards, and binds potassium that is outside of the cell and pushes it inwards. And this pump moves sodium down against its concentration gradient from inside the cell to outside of the cell, and potassium against its concentration gradient from the outside of the cell to the inside of the cell. And so this also uses ATP. And the sodium potassium pump is really important because it helps to maintain our electrochemical gradients that we have on muscle fibers as well as on our nervous cells, our neurons. So without this membrane, this polarized membrane, our cells wouldn't fire properly and we wouldn't get any signals. And so this is how sodium potassium pump works. When sodium potassium pump is in its resting form, it's open to the inside of the cell. And so sodium, 3 sodium ions can bind. When they bind, ATP can bind, and when ATP binds, it activates the pump. So it gets hydrolyzed to ADP plus the phosphate. The pump gets activated and opens up to the extracellular region. Sodium is released, but now potassium can bind. Potassium binds, the phosphate leaves the pump, and when it leaves, the pump goes back into its resting configuration, and the potassium can move into the cell. Secondary active transport uses energy of ion gradients. So you're moving molecules against their gradient using another molecule that's going down its gradient. So it's like one, it would be like sodium channels opening and sodium rushing down its concentration gradient. And as it's rushing down, something else is moving out. Or sodium is rushing down and something else is going to just jump onto the kinetic energy that's driving the sodium down its concentration gradient and ride in with it. So we have symporters and antiporters. Symporters move two substances in the same direction. One of the substances will be going from higher to lower concentration gradients using facilitative diffusion. The other substance is going to be going against its concentration gradient, but it's going to move in conjunction with the ion that's moving down its concentration gradient. Antiporters move molecules in opposite directions. So sodium, again, typically it's always sodium. Sodium is moving down its concentration gradient, and then hydrogen ions jump on the kinetic energy, and they move in the opposite direction out of the cell from lower to higher concentrations. And so here we have sodium ions and glucose. Sodium moves down its concentration into the cell, bringing glucose in with it. So here's another sodium channel, and sodium moves down its concentration gradient. As it moves, hydrogen ions jump onto that bandwagon and they move out of the cell against their concentration gradient. The last form of active transport that we talk about is vesicular transport. So vesicular transport uses vesicles, membrane-bound vesicles, so vesicles that are part of the plasma membrane, to either move things into the cell or release things out of the cell. Exocytosis, we're exiting the cell. So molecules are moving from one side of the cell to the other, from inside to the outside, and we're opening up this vesicle and molecules can be released. In endocytosis, the plasma membrane engulfs something and pulls it in. So you're bringing in a vesicle versus having a vesicle fuse and open up and then release the substances. So exocytosis is how we release proteins or other molecules from the cell that the cell's made that need to be released, or we release waste products that can't move across the plasma membrane. So the vesicle that is produced inside the cell binds to the plasma membrane, and the vesicle then opens up, releases its contents, and the vesicle becomes part of the plasma membrane. So here's exocytosis. Here's a vesicle that was formed inside the cell. It's going to bind to the plasma membrane. It's going to use the energy of ATP to open, and it releases its contents, and now we have a plasma membrane that's a little bit longer than we had before. Endocytosis is then the taking in of substances. So the plasma membrane actually becomes the vesicle for whatever we're taking in. There are three types of endocytosis that we talk about: phagocytosis, Pinocytosis, and receptor-mediated endocytosis. Phagocytosis is basically cellular eating. We're taking in large molecules. The best example is your white blood cells engulfing bacteria, pulling the bacteria into the cell, and then degrading the bacteria. So as we phagocytize the material, it moves in. The vesicle that we produce from the plasma membrane enters the cell and fuses with a lysosome. The lysosome then can release enzymes that will degrade the cell. And then the materials that are left over can actually be released through exocytosis to get rid of the waste. So here is endocytosis, a large molecule being taken in. The plasma membrane actually forms around it, forming like pseudopods around it, and then pulling it in. Pinocytosis is taking in of small droplets. So all of our cells in general can form pinocytosis, whereas phagocytosis can be only performed by certain cells. And so this is how we take in like droplets of, so it's thought of as cellular drinking. And we take in droplets that have dissolved molecules in it. And so here we have pinocytosis. Most of our cells can do this. They just take in a small amount of the fluid in the interstitial region, and that blebs off, and we now have this vesicle which has some certain molecules in it. These molecules could be beneficial to the cell, or they might be something that we want to degrade. It just depends. Then we have receptor-mediated endocytosis. And so this is a very specialized form of endocytosis in which we can take in certain types of molecules in bulk quantity. So we take in a lot of them at one time. This is how we get rid of cholesterol from our blood. So when you have cholesterol in your blood, it can cause all kinds of problems. You know, LDLs can cause high blood pressure. They can block your arteries, leads to heart attacks. It can lead to strokes. So there's a lot of problems with having a lot of cholesterol in your system. So what we have is we have these special proteins that are embedded in certain cells. And these proteins have this special coating called clathrin. This clathrin is coated around the vesicle. And then we have these receptors for the cholesterol. Cholesterol binds to each of the receptors, and then once all of the receptors are bound, then all the cholesterol is brought in to the cell. And then the cholesterol can be broken down, or it can be reused from other molecules, whatever we need to do. And I think, yep, I'm gonna stop here, but I want to just mention that this receptor-mediated endocytosis is really important because it's how we get rid of our cholesterol. And so you've heard of people that have really high cholesterol. They have diseases associated with their cholesterol. What the problem is their genes, so the proteins that make up these receptors are faulty. And so some people can have two bad genes where all these receptors are not going to work properly, or they can have one bad gene where a few of the receptors are faulty, but others will take in cholesterol. And most people don't have faulty genes, so that's good. You know, we're pretty healthy in general when it comes to cholesterol if we're eating a healthy diet. If you have this disease and you have these receptors that don't function properly, then you're not going to be able to pull the cholesterol out of your blood, and that can lead to severely high cholesterol levels, even if you don't eat a lot of cholesterol. So my husband has this disease, and his cholesterol, if he's not taking his medicine, can be up above 400, above 600. Two of my children have this disease. They received the bad allele from my husband. And so all three of them have the disease, but they have one normal gene and then one faulty gene. And so two of my four children have this disease, and they also have high cholesterols, above 300 when they were 11 and 12 years old when we found out. So that's really atypical of young children. but they had it because they have this disease. And so they also are on medication at this point. I'm going to stop here. This is a little longer than normal. We'll start with our organelles in the next lecture. Bye.